Hello everyone, myself Dr. Deepika Saini, Associate Professor at Lloyd Institute of Management and Technology, Greater Noida. As in the previous lecture, we have discussed the initial of sedative and hypnotic in one of the important class, benzodiazepine. In today's lecture, we will discuss the further classification of sedatives and hypnotics, including barbiturates, its structure activity relationship, important synthesis, and along with the miscellaneous agents. So, let us begin with barbiturates. Barbiturates, all barbiturates are the derivatives of barbituric acids. They act as central nervous system depressant and they effectively act as anti-anxiety, as hypnotic, as anticonvulsant as well as analgesic. They have a very addiction potential in case of both physical as well as mental dependence. Because of this property, benzodiazepine have largely replaced them when we talk about sedatives and hypnotics. So, first of all, we will study about the different types of barbiturates. Barbiturates are further classified as per their duration of action they spend in the body. First of all, long acting barbiturates. Long acting barbiturates means they will have 6 hour or more than 6 hour duration of action in the body. Examples are barbital, phenobarbital and mephobarbital. And this is a general structure of barbiturates. Here we start the numbering from nitrogen, this N1, 2, 3, 4, 5 and 6. There are substituent possibilities at position number 1 at the nitrogen and position number 5. There are 2 hydrogen atoms that can be replaced. So, we have written R5 and R5 dash. So, these substituent can be similar and can be different. Like in case of barbital, this nitrogen is not substituted, this is unsubstituted, here is hydrogen. At the position number of 5, we have 2 ethyl group when we talk about barbital. In case of phenobarbital, it is almost similar to barbital, the only point is one ethyl group has been replaced with a phenyl ring. Further, mephobarbitals. In case of mepho, as we know, mepho represent CH3, the methyl group. So, when this nitrogen is substituted with the methyl group, so it is known as mephobarbital. It is almost similar to phenobarbital. The only difference is nitrogen is being substituted with a methyl group. Further, we have intermediate acting barbiturates that will stay in the body, that will have a duration of action of 3 hours to 6 hours. The example is amobarbital and butabarbital. Amobarbital, in this case, again the nitrogen remain unsubstituted in both of the cases, but when we talk about the phenyl ring, we have 2 different substituents. In case of amobarbital as well as in case of butabarbital. In case of butabarbital, we have the 2 substituents here and we have butyl substituent here. It is also, this is the example of amobarbital and butabarbital. Next is the short acting barbiturates. Short acting barbiturates are those barbiturates that will spend less than 3 hours in body for their action as sedative and hypnotic. The example is pentobarbital and secobarbital. In this case, the nitrogen again remain unsubstituted, the substituent at position number 5 varies, like in pentobarbital it is C2H5 and a isopentyl group at position number 5. And in case of secobarbital, here we have double bonded substitution in case of secobarbital. Next is ultra short acting barbiturate, that stays for 15 minutes. And when we talk about this is thiopental sodium. This is an example of barbiturate category, but it is not under sedative and hypnotic. It is an anesthetic compound, so it will produce anesthesia. Now we will talk about mechanism of action of barbiturates. When we talk about how barbiturates act, these drugs increase the lifetime of calcium channel opening induced by GABA. Means again they will enhance the activity of GABA. GABA induces the calcium channel opening and lead to depolarization and hence CNS depression activity. At very high concentration, barbiturates depress the voltage sensitive sodium and potassium channel also, and they also block the excitatory glutamate receptor. This glutamate receptor is responsible for the excitement. By blocking this glutamate receptor, they decreases the excitement and hence act as sedative and hypnotic by inducing calmness in the recipient, in the patient. Now we will talk about structure activity relationship of barbiturates. To understand the structure activity of barbiturates, first we have to understand what is the requirement to possess hypnotic property. When we talk about barbituric itself does not possess any hypnotic activity. By substitution, by various substitution at various position of barbituric acid, that may lead to the production of compounds that possess hypnotic activity. When we talk about a hypnotic activity in case of barbiturates, like in barbituric acid, it does not possess any hypnotic activity. It should have a very right balance of acidic as well as lipophilic property to be an active compound for hypnotic and sedative activity. When we talk about to be acidic or to be lipophilic, so first acidic. When we talk about to make a barbituric acid derivative acidic, then we should keep this in mind, either both or at least one of the two nitrogen atom that must remain unsubstituted. Means the nitrogen, like we have two nitrogen atom, 1 and 2, position number 1 and position number 3. Either both remain unsubstituted or at least one nitrogen remain unsubstituted to possess the acidic character that is required for the hypnotic activity. The second thing that we should keep in mind is the lipophilicity. And when we talk about lipophilicity, then both hydrogen atom at position number 5 must be substituted. When, like we have seen, there are two hydrogen like R5 and R5 dash, we have two substitution options. These two hydrogens, the hydrogen which is being replaced by any substitution. So the hydrogen, both the hydrogen at the position number 5, they must be replaced by any appropriate alkyl or aryl group. We can use alkyl like ethyl, methyl group, or we can also use phenyl derivatives. So here are few examples, like first of all, when we talk about barbituric acid, why is it inactive? Because we have seen, we have talked about the acidic property and the lipophilic property. When we check the acidic property of barbituric acid, it has two unsubstituted nitrogen atoms, means surely it possesses acidic property. When we talk about the lipophilic character, the position number 5 that contains two hydrogen atoms must be replaced by an appropriate substituent. That has not been happened. So, this is not lipophilic, means it is inactive. Further, we have elucidated this property by giving several examples, like in structure number 1, when we talk about this structure, here the compound has same problem as that of barbituric acid, only because these both nitrogen are unsubstituted. Unsubstituted means that is 100% surely it's an acidic compound, but when we check the lipophilicity, it fails. It is not lipophilic because both of the hydrogen must be substituted. Here only one substitution means it contains one hydrogen atom. So, it is not lipophilic as it is not active in case of hypnotic activity. Further, similarly, when we see this example, in this example what has happened, now lipophilicity is perfect because the both of the hydrogen atoms has been replaced by a particular substituent. But now when we check the acidic character, see here both of the nitrogen has been substituted, which is not a good sign for the activity of hypnotic property. So, this is not active, this is inactive. Now, see the example of this A and B, these two compounds. Very good, both the substitution at position number 5 means lipophilic, and at least one nitrogen atom is unsubstituted, at least one nitrogen atom. We will prefer both the nitrogen atom to remain unsubstituted, but if at least one nitrogen atom is unsubstituted, means that possess acidic character. So, this possess acidic character as well as lipophilic character, means this compound A has maintained the balance between the acidic as well as lipophilic character, hence it is active compound. When we talk about the structure number B, it has the similar characteristic as it has substituted both of the hydrogen at position number 5. And when we talk about the nitrogen, both of the nitrogen that must remain unsubstituted are unsubstituted, hence it also possesses acidic character. Again, this compound B is also active. So, when we talk about these two structure 1 and 2, these are inactive because of being either not lipophilic or not acidic, but when we talk about the characteristic of compound A and compound B, they both are active, they possess both acidic as well as lipophilic character. Further, when we talk about the explore the structure activity relationship of this barbiturate category. First of all, the total number of carbon atom that is present at position number 5 is position number 5 पे प्रेजेंट रहने वाले दोनों कंपाउंड्स, द टोटल नंबर ऑफ दैट वी हैव टू कीप दिस इन माइंड, द total number of carbon atom that is possessed by both of the substituent. When we talk about, like suppose if both are substituted with ethyl, so the number is 4. Fine. So, we must keep this in mind, that number should lie in between the range of 4 to 10, means this compound must have at least ethyl substitution. It is not possible like one is ethyl, another one is methyl. So, the total number of carbon atom is 3, that is not allowed. So, the total number of carbon atom present in both groups at carbon number 5 must not be less than 4 and more than 10. It will affect the onset of action and the duration of action as shown in this table. Like when we talk about the 4, it means when both the R5 and R5 dash position is substituted with ethyl group. Ethyl means total number of carbon atom is 4. So, that will have slowest onset of action and longest duration of action, that will stay for a longer period of time in the body. And when we talk about the range of 5 to 7, they will act as intermediate acting, that will have the intermediate duration of action. When we talk about the 7 to 9, they have a very shorter duration of action in the body, and that will produce its action rapidly when it has like 7 to 9 carbon atoms. So, like when we have taken the example of barbital, because in this case is C2H5 and C2H5, the shorter chain at the carbon number 5. So, that will resist the oxidation, and hence that will not get metabolized easily. And if that will not get metabolized easily, means that will stay in the body for a longer period of time. That is why barbital is a longer acting, that is a long acting. When we talk about a pentobarbital, it is long chain at the carbon number 5. This is the structure of pentobarbital. So, that will readily oxidize and hence shorter acting in case of as compared to barbital. So, in this case we have studied the total number of carbon atom that is present at position number 5. So, that must be keep in mind relating to the onset of action and the duration of action. If number is less, that will have longer duration of action. If number is large, then that will have a shorter duration of action, as we have seen in the examples of barbital and pentobarbital. So, the next is the branched chain isomers, means when we are talking about branching in this substitution, like that branched chain isomers exhibit greater activity but shorter duration of action. More is the branching, more potent is the drug. When we have compared the pentobarbital with the amobarbital, what happened here? In the case of pentobarbital, this substitution, this branching at position number, at this position number 1. So, this branching leads to greater activity as if we compare it with the amobarbital as compared to lesser branching. Next, when we talk about the double bond, how the double bond affects the activity. So, double bond in the alkyl substituent, that will produce compound that are susceptible to the tissue oxidation. If they get oxidized readily, means they are short acting. Like in the example of secobarbital, when we talk about secobarbital, here is the double bond in the substitution at position number 5 that will make it more susceptible for the tissue oxidation. That is why secobarbital, it is a short acting drug. Now, if we compare the aromatic compound with the alicyclic compound, aromatic and alicyclic moieties, they exert greater potency as compared to common aliphatic compounds. For example, even if they possess the same number of carbon atoms. For example, when we talk about the compound number 1 and compound number 2, here the only difference, they have same, both nitrogens are unsubstituted, When we talk about the other structure, this ethyl group, that is also similar. This is the only difference, that contains an aryl, that a phenyl ring, and the another one that contains a state aliphatic compound substituent with the same number of carbon atom, that is 6. Here is the 6 carbon ring and here is a 6 carbon chain. So, according to the first theory, that the number of carbon in the substitution at position number 5, there is 8, here is also 8, but there is a huge difference in their activity, because aromatic and alicyclic moieties, they possess more, they will produce more potent compound as compared to the aliphatic one. So, that is why this compound 1, that will be more potent as compared to compound number 2. Further, when we discuss the substitution with the nitrogen atom, when we talk about the substitution at nitrogen atom, so the methylation of one nitrogen atom, methylation either at this position or at this position, that enhances the onset and reduces the duration of action, that will increase the onset of action and hence reduces the duration of action. The drugs will be short acting. Example is methyl pentobarbital. When pentobarbital, one of the nitrogen of pentobarbital is substituted with methyl group, that is methyl pentobarbital, and that will possess the shorter duration of action. Now, if we are talking about the substituent at position number R5 and R5 dash, if we include any halogen, any chlorine, fluorine or bromine atom at this position as a substituent, that will increase the activity, that will enhance the activity. When we increase any kind of polar group, OH, carbonyl, COH, NH2, SO3H, we know that will reduce the potency, and apart from that, being polar, that will be more easily oxidized and will be removed from the body. So, by the inclusion of halogen, that sounds to be more appropriate as compared to inclusion of polar group at position number 5. So, when we thought of replacement of oxygen atom with a sulfur atom at the position number 2, suppose if we replace, when we have replaced a carbonyl group here with a sulfur group, with a sulfur atom. So, that will significantly increases the lipid solubility. That is why if we have increased the solubility in the lipid, means that will pass the plasma membrane and the other membranes quickly, that results in rapid onset of action, because it reaches the brain level at the maximum value. And such drug, like we have already given an example of this, thiopental sodium, it is well known as IV anesthetic. So, this is one of the important point to be noted, like when we have replaced this nitrogen, this oxygen atom with a sulfur atom, that will increase the lipid solubility, and hence that has changed the mechanism, that has changed the activity of a particular compound. Now, they will not act as sedative and hypnotic, now they will act as IV anesthetic. This is a synthesis of barbital, where the urea is treated with diethyl 2,2-diethyl malonate, and with the action of sodium ethyl ether and ethyl bromide, that will lead to the formation of barbitone. Further, with the NaOH, that converted into its salt, barbitone sodium. So, this is the synthesis of barbital. There are several miscellaneous agents, amides, imide, like example is glutathione, alcohols like meprobamate is the aldehyde and the derivative like triclophos sodium and parahaldehyde are there. So, these are the structural examples of miscellaneous agents. These are further, this is example of miscellaneous agent triclophos sodium and paraaldehyde. Thank you.